so we've got that. Let's see, do I have more sets? Oh, I do. Okay, so this is the model that I've been telling you about.
Additive.
This additive linear model. There's also a model where you can assume there's some interaction going on between your genetics and the environment. So one instance here might be, let's say you're a smoker, and there's some there's some variant in your body, and in a non-smoker, there's no effect. But if you are a smoker, then all of a sudden that variant does play a role in some phenotype you're looking at. Or
cancer.
Or cancer. Yeah, cancer is a great example of this, where there's probably a whole ton of these, we call these gene by environment interactions. So then you also have this option called a gene-gene interaction, and
that's what you were talking about.
This is what you were talking about earlier, and this is where your genotype at one SNP location doesn't have an effect, or it does have an effect by itself, but also that effect can be influenced by the genotype at a different position in the genome. And so it's just like these two SNPs now interact in individuals, and they don't do it independently.
This is also called an epistatic interaction, is that correct?
Yeah, people also call this an epistatic interaction. Sometimes you'll hear this referred to as a haplotype effect, is another term. So a haplotype effect is usually when the two SNPs interacting are on the same haplotype or very near each other, whereas an epistatic effect, they don't necessarily need to be near each other, even on the same chromosome. So they can be really far apart, but for some reason are still interacting together. Yeah, so, epistatic or haplotype.
Epistasis is also another way to say that.
Yeah, epistasis. All right, so this was the math that I showed you for this first model, and this is the math for this last model. So I'm showing you this not because you actually need to know this, so don't bother writing this down, but I'm going to walk you through it quickly so you can see how complicated it is. So this first part right here is actually the exact same thing up there, but now I put the superscript on the beta I. So this is the effect of genetics for SNP-I. Now I'm going to look at the E component. Not the E component. Now I'm going to look at the E component, is still here at the E at the end. However, now I'm going to look at these gene by environment interactions. So this beta is now beta GD, and that stands for beta genetics in diet, because somehow your diet is now interacting with your genetics. And so here's the genotype of your genetics. This ED is just some measurement of your diet. It's like, how many apples do you eat a day? Do you like tomatoes? I don't know. It's just some measurement that people came up with for your diet. And obviously they do a lot of work to get this. And so that diet plus your genetics is going to somehow interact with some effect size, beta there. Here, the same thing. Here's now your genetics. This E is S for, say, sleep or sports, I don't know. And it's going to have its own set of betas that are there. So these are two different environment by gene interactions that could exist, and you could literally have as many of these as you want for any environment you can measure. And then over here, here's the real kicker. So this is this G by G, so the gene by gene interaction. And as you can see, it's one genotype at position J, which is now not the individual, because you can use J's multiple times. So this is now at position J times the genotype at position I, so two different SNP positions, multiply them together, and then they're going to have some beta effect size, which I'm doing here is beta GGIJ.
Which is the interaction effect.
Which is the interaction or the epistatic effect or the haplotype effect, however you want to call it, between